Hello everybody, welcome to this presentation about computational analysis and modeling of neurological conditions. So I will be talking about methods, computational innovative methods that allow to analyze, study and also recognize pathological conditions of particularly the brain. And what I would like to highlight here initially is that our brain undergoes a lot of changes during the entire life. So when we are born, we all originate from, before we are born, we are all originate from a single cell, which is the zygote. And then there is gastrulation, which according to the eminent developmental biologist Louis Wolpert is the most important time of our life, even more important than birth, death, or marriage. And then there's neurulation, birth, childhood, of course, adolescence, and our brain undergoes a lot of changes during adolescence, as you will know, and then degeneration and death. And so by using computational tools, one can not only study what is going on in these transitional stages, but also use this information and those tools in order to detect if maybe some changes are different from the norm. And so one can use this, for example, to look at anencephaly or autism spectrum disorders, and also Alzheimer's disease, as I will talk to you in this presentation. So by looking at the brain, we are actually looking at a very complex organ. So the brain, the human brain, is the most complex entity known to humankind. And here you see a very nice visualization from a previous colleague of mine who created this three-dimensional visualization as obtained from diffusion tensor imaging. So diffusion tensor imaging is an MRI method that allows to measure the diffusion of water molecules, and indirectly one can use those measurements in order to infer fiber tracts in the brain. So here you see such fiber tracts where different colors indicate different orientations. So blue would be vertical orientations, and red horizontal, and green would be oblique oriented fiber tracts. And of course, there's a lot of information there, but we can infer statistically information about connections between different parts of the brain, different brain regions. And so what we did in a study a few years ago is we looked at such DTI data from the very well-established ADNI database. So the ADNI database has such information from healthy subjects, so brain connectivity information that we can infer from the data that is available on this database, but also on subjects with Alzheimer's disease. And what we did here in this study is we looked at those networks, and we then employed machine learning in order to check if there is a difference. And then if one has trained such a machine learning method, then one can use this, of course, also to diagnose Alzheimer's disease. And so what we did in more detail is we actually focused on particular brain signatures. So we looked at certain characteristics of those networks across the brain in different parts. We looked at what is characteristics in healthy subjects, and what is maybe characteristics in Alzheimer's disease patients. And what we then did is, and I consider this quite different from other approaches, is that we actually created a computational model for what is happening during normal aging. So the brain changes during normal aging. Even if you don't have Alzheimer's disease, your brain is changing all the time. And during normal aging, certain connections are decreasing in their weights, and some are decreasing more than others. And we created a model of those changes during normal aging, as well as such a model as during aging when someone has Alzheimer's disease. So there are some differences there. And we formalized that. We created a computation model that describes this for all the different connections in the brain. And then what we did is we simulated this progression across 15 different stages. So by starting at a healthy condition, so in both cases we started with the healthy conditions, and then we simulated what we expect to see happening with those connections during normal aging, as well as what we expect to happen during Alzheimer's disease aging. And we used machine learning then to differentiate between those two different progression models at each stage. And one stage refers approximately to 1 year. So then what we did is we used a so-called complex network approach. So we looked at the DTI data, and then we inferred networks from those DTI data, and then we formulated this as a graph and ultimately as a complex network, because brain networks are actually highly non-random. So if you look at the regular random network, like for example an Erdos-Renyi network, then you have very different characteristics of those networks. And so that's why we took a complex network approach. In the brain, you have certain regions that have many more connections than others. So these are so-called hub regions, and they are actually a lot more affected during Alzheimer's disease than others tendentially. And also, we looked at other complex network features like that are related to centrality of nodes, and so this is a whole field of mathematics. This is about complex networks, and we used those particular features that allow to characterize a complex network. And so, for example, here this could be, for example, a hub, because it has more connections than, for example, this node here, and so one can differentiate also individual nodes just based on their connectivity. And so what we did then is we extracted a lot of different complex network related features from each subject, and then we did feature importance ranking. So we looked at the different features and used machine learning in order to identify features which are more informative than others for classifying Alzheimer's disease versus healthy subjects. And as you can see here, there are different types of complex network features, like, for example, the strength of individual nodes connections, or their closeness. So these are all, they all tend to be quite strongly related to centrality of a complex network, and these are the network features that we identified as being very important in order to classify Alzheimer's disease. And what we also did is, as I said at the beginning, is we essentially applied this classification method at different stages. And as you can see here, we see what we would, this is what we would expect, right? We would expect that at early stages, when there's not a lot of changes, neither in the normal aging as well as in the pathological aging model, that the classifier will not have much of a difference in both cases, in both scenarios, right? So you will be close to 0.5 classification performance. And then as this increases, as the stages increase, so the more progressed Alzheimer's disease becomes, the better the classifier will be at distinguishing those two different trajectories. And this is exactly what we see. So here you see such a figure from our publication, where here on the x-axis we have the different stages of progression, and on the y-axis we have the performance indicators of the classifier, the machine learning classifier. And you can appreciate that here at around -5 or -4 stages before the end point of progression, we can already differentiate with quite high reliability and performance the two different types of brain networks. And 0 would essentially refer to a stage of Alzheimer's disease where with current tools we can well classify or diagnose Alzheimer's disease. So this figure essentially tells us that we, by using this innovative approach of modeling the progression, we can actually identify or diagnose at least an elevated risk for Alzheimer's disease before current state-of-the-art methods can do. And this is also statistically shown here. The p-value becomes very small here at -5, -4 years or stages. And so overall, this shows that by using computational methods and machine learning methods that use statistical tools, we can indeed gain information that is very valuable for the diagnosis of Alzheimer's disease just based on the structure. So here we don't look at function at all. And of course, there are ways also to extract information on the functioning of the brain, such as, for example, EEG. And we published recently, last year, we published a paper exactly on this. So we also looked at function, and so one could use different data modalities. Now, this was about analyzing brain structure in order to diagnose Alzheimer's disease, but one can actually also look at other parts of the body, in particular the retina. So the retina is a tissue at the rear of the eye, and so it is peripheral, but it is part of the brain. It is a neural tissue also, and one can very easily and quickly and cheaply image the retina. Here you see images, so-called retinal fundus images, and you can clearly see here also the vasculature. And there are computational methods in order to analyze this automatically. And this is exactly what we did a few years ago. A master student of mine has very nicely implemented some computational tools like convolutional neural networks in order to segment the blood vessels, and then he also used such methods to classify those vessels into arteries and veins, and there are different ways of how one can do this, of course. One nice thing or aspect of this kind of approach is also that one can extract features directly from the image. So this one can then interpret those features, and I will not go into detail into those features here because that would take more time than I have. But essentially, the message that I want to bring across here is that those features seem to vary in the two different conditions of healthy versus demented subjects. So you see here that the distributions are quite different, and we have also we are currently actually looking into this in more detail with a PhD student of mine. But the important is here that we can interpret and can humanly grasp or understand certain things that happen in the retina in those two different conditions of healthy and dementia or Alzheimer's disease. And this is, of course, very important in a biomedical setting, right? We don't want just to have a classifier that tells us that someone has Alzheimer's disease without knowing why. And so it is very important to be able to interpret and explain the reasoning behind such classifications, and that's exactly what we are focusing on. And we have actually published some of our work recently, and here we used a slightly different computational approach. We used so-called inductive logic programming, or ILP, and this is a symbolic AI approach. And the one advantage that we have here, which is a very big advantage in a biomedical setting, is that we don't need a very large number of data sets or samples in order train properly these models. So what you see here is on the x-axis the number of negative examples, and with our method we can actually very well we improve consistently our performance or accuracy with ILP as compared to using deep learning, state-of-the-art deep learning methods, because ILP is a different approach of AI. It can actually exploit a smaller number of samples, and this is of course important in a biomedical setting, because not only are you often limited in terms of the samples from subjects with a certain condition, but also subjects vary, of course, also depending on their age, or they might have comorbidities. And so by using ILP, we can get around this significant problem. And this brings me to another project that I'm involved in, which is Biodynamo. So I'm a co-founder and also I'm the spokesperson of the Biodynamo collaboration, which is an international collaboration involving multiple very prestigious institutions. And we have created a software that allows to simulate biological systems, including neural systems. So we can simulate neural networks and brain tissue across time at very high detail. So we can really model connections and axons and dendrites over time and their interactions. So this is a different approach to what I showed you before, where we essentially look at the graph of the brain. Here we really model the tissue on a cellular level. So we really model individual cells and cell compartments. And so what you might find interesting is also that we have published this and also provide this as open source. So Biodynamo is available as open source for free for the community. Feel free to have a look at the website. We have the source code that is available on GitHub. There is installation guides, tutorials, examples, demos, and so on. There is also a forum where you can ask questions. And we also have an active community of developers and users of Biodynamo with regular meetings. So if you're interested, please feel free to have a look here and get in touch with me. I would be happy to advise you and respond to any queries you may have. But going back to the medical or biomedical significance of this, we can use Biodynamo now to simulate brain tissue, or for example, as in this case, we can simulate also brain cancer. So we have here in this case simulated glioblastoma in 3 dimensions and how it changes over time. So the progression of glioblastoma. And as you can see here, we can look at various parameters or variables of these systems, for example, oxygen saturation or the density of certain cell types, because glioblastoma includes different cell types. And so we can really model how over time these changes, which can be very helpful and which will be very helpful for a personalized medicine approach, because each subject has different conditions, of course, and different genetic predispositions. And by using this kind of in silico approach, we can better understand the progression and what kind of treatment might have the best or biggest impact on the progression. And of course, for that, we need also a lot of data. And so this is currently an ongoing research focus of my lab, is to better understand how to bring in this data and how to use those for predictions and potentially also treatments ultimately. And an important advantage of this is also, of course, that we can infer certain information that we cannot do experimentally, or which would be very difficult experimentally. Here we can really understand what we expect to see in 3 dimensions at a very high spatial and temporal resolution, right? You cannot just from a subject who has cancer extract tissues, especially in the brain, like that. But here we can model this. And of course, we need also experimental data in order to iteratively refine those models in order to improve their accuracies. So to summarize, I would like to highlight that that if we are interested in using interdisciplinary approaches, especially also for biological and biomedical, in a biomedical context, we need to appreciate that the brain and our bodies overall follow trajectories. They're always changing, and AI and machine learning can be employed to study those systems, to analyze them, but also to model them. And we can do this using data from the brain, but also, for example, from other systems like the retina, as I showed before. But of course, you could also look at blood markers, for example. And there is, of course, also research going into this direction. But the important point here is that the indicators don't need to be necessarily directly from the brain. They can be also from other parts of the body. And there are different computational approaches. There we can use statistical machine learning, as I showed at the very beginning, with those complex brain networks. We can use symbolic AI, as the ILP example showed. And we can use mechanistic models like this agent-based software Biodynamo, which really models on an individual cell basis the entire system. And so there is a lot of research questions that we can address along those lines, not only with regards to the experimental side, but also to the computational side. And so this is I think, a very fertile interdisciplinary research direction. And so I would like to thank you for your attention, and I would like to thank my collaborators. I've listed some of them here. I cannot thank all of them on one slide, but I'm very grateful for all the many contributions from my colleagues, and I of course want to thank also the funders who have made it possible to do this research and to follow this research approach, and my lab, which is a very lively bunch of lab members. And of course, if you have any questions, please feel free to email me. This is my email address or my Twitter feed. Feel free to follow me. Thanks for your attention.